Welcome to video series on acid-base balance. Here is a quick summary of metabolic acidosis. The main biochemical basis for metabolic acidosis is deficiency of bicarbonate, or it can also be because of increased production of acids or reduced excretion of acids. There are two types of metabolic acidosis: high anion gap metabolic acidosis and normal anion gap metabolic acidosis. The causes are for high anion gap metabolic acidosis are as follows. The first cause is renal failure, characterized by impaired excretion of hydrogen ions. So these hydrogen ions accumulate, resulting in high anion gap metabolic acidosis. Next is severe exercise, characterized by anaerobic glycolysis because the cell shift to anaerobic glycolysis, resulting in increased production of lactic acid, leading to development of high anion gap metabolic acidosis. Among chronic alcoholics, what happens? Alcohol gets oxidized to acetaldehyde and NAD gets reduced to NADH, and this will in turn reduce pyruvate to lactate. So lactic acidosis sets in chronic alcoholics, leading to the development of high anion gap metabolic acidosis. The next cause is diabetic ketoacidosis, where there is relative deficiency of insulin, characterized by hyperglycemia. As we all know, insulin is an anabolic hormone. With deficiency, catabolism of fatty acids occurs, which are oxidized by beta oxidation, and this will end with formation of increased acetyl CoA, which gets diverted to form ketone bodies like acetosic acid and beta hydroxybutyric acid. They accumulate and lead to the development of high anion gap metabolic acidosis. So these are the four important causes for high anion gap metabolic acidosis. You can remember the mnemonic READ, READ, which stands for renal failure, severe exercise, alcohol, and diabetic ketoacidosis. Coming to normal anion gap metabolic acidosis, the causes include carbonic anhydrase inhibitors, drugs like acetazolamide, which will inhibit the formation of bicarbonate in the renal tubular cells. As we all know, the metabolism of renal tubular cells lead to production of carbon dioxide, which will combine with water to form carbonic acid under the influence of carbonic anhydrase. Now this carbonic acid dissociate to form H plus ion and bicarbonate ion. So drugs like acetazolamide or carbonic anhydrase inhibitor will inhibit the generation of bicarbonate. So this contribute to the development of normal anion gap metabolic acidosis. Here, in all these conditions, the bicarbonate is decreased, and as a compensatory mechanism, chloride is increased. So in all these conditions, it is characterized by hyperchloremic acidosis. The next condition is ureterosigmoidostomy, the procedure where urine is diverted by anastomosing both the ureters into sigmoid colon. So the urine is lost through intestine, resulting in large amount of loss of bicarbonate. So the same thing happens in different types of intestinal fistulas, characterized by loss of bicarbonate. So next one is renal tubular acidosis, characterized by defective reabsorption of bicarbonate in proximal convoluted tubule, leading to development of normal anion gap metabolic acidosis. The most important cause actually is diarrhea, severe diarrhea, where the bicarbonate is lost through intestinal secretion. So in all this secretion, as a compensatory mechanism, chloride is increased. So in all these conditions, they are characterized by hyperchloremic normal anion gap metabolic acidosis, and the mnemonic to remember this is CURD, C-U-R-D. C stands for carbonic anhydrase inhibitor, U for ureterosigmoidostomy, R for renal tubular acidosis, D for diarrhea. Coming to ABG analysis, pH is decreased, bicarbonate is decreased, partial pressure of carbon dioxide is normal. So metabolic acidosis is characterized by hyperkalemia and hypercalcemia. Hyperkalemia because there occurs redistribution of hydrogen ions in exchange for potassium ion. So metabolic acidosis is characterized by increased hydrogen ion concentration. So this moves from the bloodstream into the cells, and in order to maintain the electrochemical gradient, potassium ion moves out of the cell into the blood, resulting in hyperkalemia. Now, metabolic acidosis is also characterized by hypercalcemia. This is because the calcium is the normal binding site for all the cations and anions. In metabolic acidosis, there is increased concentration of hydrogen ion. So this will go and bind to albumin, displacing calcium from the binding site of albumin, and this calcium enters bloodstream, resulting in hypercalcemia. Now, how is this metabolic acidosis compensated? So there are two important compensation rules. Compensation always involve the counter variable, but in the same direction as that of the primary variable. Primary variable in metabolic acidosis is bicarbonate, and this is decreased here, and the counter variable is carbonic acid. In metabolic acidosis, the counter variable is carbonic acid. Since the primary variable is decreased, the counter variable has to be decreased in order to attain the compensation. Now the carbonic acid concentration is decreased by hyperventilation by the lungs and by excretion of hydrogen ion titratable acid and excretion of ammonium ion by the renal system. So this was all about a quick summary of acidosis. Next video will be on metabolic alkalosis. Thank you. Please do like, share and subscribe. Thank you.